Good afternoon, everyone. As Bob mentioned, I'm glad to present this webinar about the REPROCELL Biobank, which is a unique tool for neurodegenerative disease applications. So first, I want to give you a little bit of background on why we are relevant in this field and which skills we are bringing to the table. So in 2014, REPROCELL, which is headquartered in Japan, acquired Bioserve in the US and merged it with StemGen to add stem cell products and services, and it formed REPROCELL USA. Bioserve has been maintaining a repository of over 600,000 varied samples, such as serum, FFPE, frozen tissue, and DNA or RNA, and he's specializing in custom collections as well. The merge with StemGen added all these skills to stem cell services, and it makes it easy to understand how REPROCELL is competent to run successfully a biobank centered on induced pluripotent stem cells. At the same time, REPROCELL has always been focused on neurosciences, and everyone in the neuroscience field knows that the availability of adult tissue and cells is limited or non-existent. In addition, animal models have their own set of limitations. Inside the neuroscience field, we know that most neurodegenerative diseases do not have satisfying treatment options, for example, for multiple sclerosis or MS or Parkinson's disease, or have a defined cause, such as for amyotrophic lateral sclerosis or ALS or Alzheimer's disease. This is where iPSC technology shows its power and offers a novel avenue to not only study the mechanism of disease, but also helps with drug screening. As we all know, collecting good data is critical, and having the proper set of controls is as critical to be sure that the data collected is strong and relevant. This is why we started building this biobank with a strong set of controls in mind. So we collected tissue from 8 different healthy individuals of various race and sex and age. Age was critical as we want to address needs in late onset neurodegenerative diseases. So having controls that were 45 years of age and older was high on our priority list. Also, I would like to attract your attention to 2 lines, SK003 and BL003, which have been derived from the same patient, but from 2 different tissues of origin, whole blood for BL003 and dermal fibroblasts for SK003. So that may be important for some of your research. Now, I want to introduce you to the list of our neurodegenerative disease donors and iPS cell lines available. You may notice that the list is actually longer than the one on our website. I really want to emphasize that this biobank is still growing, that we're still screening donors, collecting tissue, reprogramming, and finalizing quality control. So we started screening donors with Parkinson's disease. So this is why we have 3 iPSC lines available so far that are related to Parkinson's disease. One of the donors has sporadic Parkinson's disease, SK007, which we will talk about more during the presentation of the showcase study. Then we have 2 patients with familial forms of Parkinson's disease, one with a mutation in the Parkin2 gene, and the other one with a mutation is the SNCA gene. We procured tissue from 2 ALS patients affected with the familial form of the disease with mutation in the FUS gene and the other one in the TDP43 gene. Finally, we have 1 iPSC line from a patient with a familial form of Alzheimer's disease with a mutation in the presenilin 1 gene. So this biobank has been established with certain common characteristics. For example, we have 4 types of starting materials that we consider, such as fibroblasts from skin punch biopsies, endothelial progenitor cells from whole blood, urine-derived cells from urine, which are actually from the kidney, and the dental pulp from the tooth. The only reprogramming technology that was used to establish this biobank is our stem RNA third generation kit. As most of you know already, this technology is solely based on RNA reprogramming and thus is non-integrative, eliminating the need to screen later stem cell cultures for the non-expression of the reprogramming factors. In addition of being faster, in our hands, the RNA reprogramming gives rise to stem cell clones that are expressing stronger stem cell markers than the clones generated with other reprogramming technologies. We recommend the use of NutriStem for the culture and maintenance of the iPSC from our biobank, as it is a rich feeder-free medium that helps diminish the spontaneous differentiation. Regarding culture matrix, we are using iMatrix 511 for the reliability of the results and also for the ease of use compared to other commercial matrices. The culture, then we are using NutriFreeze D10 as cryopreservation medium. The culturing protocol and thawing protocol that we do recommend with the use of the IPS lines of this biobank can be obtained through our customer service. So all these cell lines go through a thorough QC process before being made available to our customers. The primary cultures are all screened using a viral pathogen panel that includes HIV 1 and 2, HBV, HCV, and HTLV 1 and 2. If any tissue tests positive, it is immediately discarded. The IPS cells are also screened at the time of freezing for mycoplasma, bacteria, and fungus contamination. The STR analysis at the time of freezing needs to be consistent with the genomic profile of the tissue of origin. We are also checking the karyotyping via G-bending on 20 clones. We are assessing pluripotency with immunocytochemistry on 4 stem cell markers, OCT4, SSEA4, NANAG, and TRA160. So we are checking that the free thaw viability is over 80%, and also whole genome sequencing is available if you need to have access to it. So concerning the use of the cells, all the patients and donors have been properly consented prior to tissue collection. And in the cases where we obtained primary cultures from another biobank, the appropriate MTAs have already been put in place. So for research use, these IPS cells are for R&D only for the end user. And the commercial use of the cells in the biobank is available for a flat fee. So, of course, this biobank is also being developed with neural differentiation in mind. And so we are actively developing our neural stem cell offer around the lines from the biobank. So here I am presenting some data on 8023G to show how we can increase the amount of PAC6 expression in our neural stem cell culture, and that we are focusing on our healthy control NSC offer at first, with SK003 being available right now. Alongside this effort, we have developed a related named Repro NSC, which is composed of a basal medium and 2 supplements. So for all other projects, we're also accepting custom neural differentiation projects, which can start at the screening of patients, but also involve differentiation into NSC or dopaminergic neurons, as I will present during the showcase study. We have also developed robust motor neuron differentiation protocols and astrocyte differentiation protocols, both of them making co-culture systems available for our customers. As I mentioned before, this biobank is still evolving and growing as we're continuing to screen patients, collect tissues, and reprogram primary cultures. So please come back for more updates, as we will include to our current list lines from patients with new pathologies or new mutations. Also, do not hesitate to contact us if you have any questions or if you need help or to know what is new. And we may also assist you with custom collections. So now I will present you our showcase study on Parkinson's disease. So we wanted to put together an exemplar showcase of our capability. From patient screening to tissue collection, primary culture derivation, iPSC generation, and dopaminergic neuron differentiation and maturation, and even co-culture. Of course, a typical project is totally customizable and doesn't need to go from this beginning and this end, and is completely customizable to the client's needs. Here, we will mature the dopaminergic neurons and assess their functionality via RNAseq analysis and multi-electrode array analysis, or MEA. So as I mentioned earlier, this showcase study is based on SK007, one of the line available through our biobank. So a sporadic Parkinson's disease patient was identified and properly consented. The clinical data collected was anonymized, and the skin punch biopsy was collected, from which a fibroblast culture was derived. You can see a phase contrast image of the SK007 fibroblasts in culture. The QC was performed on the primary culture. First, the genetic profile was obtained through STR analysis alongside the viral pathogen testing, sterility testing, and mycoplasma testing, as I showed before in more detail. We confirmed that the karyotype was normal, and for this project only, we performed some immunocytochemistry and confirmed with vimentin staining that our culture consisted only of fibroblasts. We then reprogrammed the fibroblasts using our stem RNA third generation kit. And you can see in the phase contrast image that at day 7, 3 days after the last transfection for fibroblast reprogramming, we already see the emergence of iPSC colonies. The iPS clones went through a rigorous QC step, including the confirmation that the STR is consistent with the one obtained from the parental fibroblast line. We confirmed that the karyotype of the clone selected was still normal, and the immunocytochemistry was successfully performed on the 4 stem cell markers, OCT4, SSCA4, NANOG, and TRA160. Finally, we also did a trailing edge differentiation assay using the human PSC scorecard assay from Thermo Fisher Scientific. And you can notice that our cell line has the potential to differentiate into any of the 3 germ layers. I also want to draw your attention to the fact that our stem cell line in the first column of the assay exhibits a stronger expression of stem cell markers than the average panel of the assay, which consists of a mix of ESC and iPSC. Following this, we initiated differentiation of the Parkinson's disease line alongside a control line using a protocol adapted from the one published by Cricks and al. In Nature in 2011. So we chose 7713G as the control line to be differentiated. This protocol gives rise to dopaminergic neuronal progenitor cells around day 21, but the neurons do not mature fully as dopaminergic neurons before day 60. For our showcase study, we decided to compare the ability of our MQ medium to mature neurons versus B27 neurobasal medium, which is a typical dopaminergic medium, and versus a co-culture with astrocytes, which is known to accelerate and deepen the maturity of neuronal networks. These three conditions are the ones we will assess via RNAseq and MEA analysis. You can notice in the immunofluorescence images below that our dopaminergic neurons, so at day 65, are a homogeneous population expressing the neuronal markers TUJ1 with very few glial cells present and expressing the dopaminergic markers TH and PITX3. So first, I will start with the RNA-seq analysis. As I just said, so we wanted to assess MQ medium compared to co-culture astrocyte neuron. So we had to differentiate a second control line, 8023G here, into astrocytes. The astrocytes were matured until day 95, and their astrocytic identity was confirmed with GFAP and CD44 staining. So because of the nature of RNA-seq, we needed to collect a pure population of neurons for the analysis, and thus a physical astrocyte neuron co-culture was not possible. To circumvent this difficulty, we set up a transwell experiment with astrocytes in the transwell. Then we were able to collect our three different conditions without interference of glial cells. When we looked at the result of the analysis, first with the Pearson diagram on the left, it is striking that the genetic background of the cell line, meaning control versus Parkinson's disease, is a bigger factor of difference regarding gene expression than the actual method of maturation of the neurons. In addition, when comparing the differences between the methods of maturation using the Venn diagram on the right side, the number of genes in common is extensive, with only less than 4% being divergent in terms of expression, sorry. The second way we compared the three maturation methods was using multi-electrode array, or MEA. In this context, we were actually able to use a true co-culture astrocyte neurons. And we performed one recording a week from week 1 after plating until week 12. For those of you not very familiar with this technique, we plated the cells onto a well that has electrodes at the bottom. And here it's 16 electrodes in our present case, which will record the electrical activity of the neuronal network directly on top of each electrode. So before week 3, we did not record any spontaneous firing that was out of background level. Starting at week 3, we can notice an increased electrical activity into the wells with MQ medium and the co-culture, and not all electrodes are firing. And we don't see all this electrical activity in the well with the dopaminergic dopaminergic medium. So each green line represents a spike of activity over time, and each graph represents one electrode recording over time. So the results at week 11 are even more striking. Here the data is presented as raster plots, where each line represents the recording of one electrode, and each group of 16 in the y-axis represents the recording of one well over time in the x-axis. So the dopaminergic medium at the top only created random spiking in the culture. The co-culture network at the bottom is spiking a lot more frequently, and strong electrical activity can be noticed in a lot of electrodes, but no synchronicity is present. However, when we look at the MQ medium well in the middle, we immediately notice the strong electrical activity on most electrodes. In addition, the spikes are synchronous in the majority of electrodes, which is called bursting. Bursts are characteristics of mature functional neuronal networks, which confirms the advantage of using MQ medium for maturing neuronal cultures. So in summary, I hope I demonstrated that the REPROCELL Biobank is a powerful tool for research on neurodegenerative diseases. Please keep in mind that it is an evolving effort that will keep growing. And I hope our showcase study provided an example of how our team can help you achieve your research goal. Finally, I would like to thank TEDCO and the Maryland Stem Cell Research Fund for awarding us a grant, which was instrumental in initiating this effort. So I hope you enjoyed the webinar.